I'll be talking about the reactivity-informed pharmacophore editing and biological evaluation of andrographolide and its A-ring analogs, and also going into a bit about one of the pathways from another paper. And so I guess as a quick brief, I'll start off by talking about the compound of interest, andrographolide, and then talk about the C19 analogs that we've synthesized, and in addition to the biological experiments that we've conducted, and then talk about our current efforts towards producing the Oxetane moiety on andrographolide. So andrographolide is a natural product coming from the plant Andrographis paniculata, and is known to have many anti-cancer capabilities. In terms of its structure, it's composed of 3 main rings: the C-ring butanylide, the B-ring exocyclic olefin, and the A-ring 319 diol system. It is mainly known to exert its biological activity exert its anti-cancer activity through the inhibition of NF-kappa B, which is a transcription factor at a crossroad of a myriad of cell signaling pathways. Essentially, NF-kappa B modulates various genes relating to cell signaling and metastasis. And so I guess stepping back, our lab has focused a lot, or many projects in our lab has focused on natural products, whether it be through the biological evaluation of or their synthetic complexities. And so much of this inspiration actually comes from this statistic of FDA-approved drugs, in that about 4% of FDA-approved drugs are natural products, and around or over 20% are actually the natural product derivatives. And so clearly there's much benefit to taking a natural product and making key modifications such that, or making that compound more potent or less toxic, and essentially improving its medicinal value. And so I guess on the right side, you can see some examples of cancer therapeutics that are inspired by natural products, with the natural products on the top and an arrow pointing down to the FDA-approved drugs and highlighting the key modifications that have been made. And so specifically with andrographolide and this study, a previous study from a group in Thailand had andrographolide, or these analogs. They had synthesized these 2 analogs of andrographolide, the trityl andrographolide and the TBDPS epoxy andrographolide analog. And they had interestingly found that these 2 analogs actually inhibit the WNT1 or Wnt1 signaling pathway in beta-catenin. And essentially, I guess a little bit about this pathway. Essentially, the activation and nuclear translocation of beta-catenin triggers the TCF and LEF transcription factors, and that directly modulates several downstream target genes that are associated with cell survival and proliferation, differentiation, and things like that. And GSK3-beta, which is a key kinase that is part of the beta-catenin, I guess, destruction complex. It essentially phosphorylates beta-catenin and tags it for ubiquitination and later degradation. And so essentially, that study had shown that the TBDPS epoxy andrographolide analog actually functions through GSK3-beta inhibition, preventing the phosphorylation and leading to beta-catenin stabilization, preventing its entrance into the nucleus. And on the other hand, the trityl analog of andrographolide actually functioned independent of GSK3-beta. So essentially, we found this interesting, and that allowed us to ask the question of whether or how large hydrophobic groups at the C19 position of andrographolide affect or change the biological mechanism of action through which those compounds or those analogs exert their anti-cancer capabilities. And so here is the library of 12 analogs that we've synthesized, highlighting the trityl ethers on the top and the silyl ethers on the bottom. In terms of synthesis, it was quite simple. The trityl ethers just required, it was a reaction with pyridine and the corresponding trityl chloride. And then to evaluate the effect of various alkyl substitutions or the presence of a methyl group, steric bulk, and the addition of a chlorine atom, we synthesized these few silyl ether analogs with the corresponding silyl chloride or triflate, noting that triflates were generally more reactive than the chlorides. And so with these analogs in hand, we wanted to test these compounds' activity or anti-proliferative activity. And so we chose four different cell lines, breast and colon cancer. We chose HCT116, A229, MCF7, and MDA-MB231. And we performed this experiment at 24 and 96-hour time points. And just to highlight a few points or a few takeaways from this study or from this experiment, majority of these analogs generally were more potent than the parent compound andrographolide across both time points and the 4 cell lines, and also amongst the 4 trityl analogs that we synthesized. The compound 5 with the chlorine substitution functioned much better or was much more potent than the other 3. And overall, compound 13 exhibited, I guess, about 1 to 2 orders of magnitude greater potency compared to the parent compound. And also importantly, all of our compounds generally had lower IC50 values in the colon cancer cells as opposed to the breast cancer cells, showing some level of cell line selectivity. And so in order to investigate the ability of our compounds to inhibit NF-kappa B, we performed the secreted alkaline phosphatase assay, or SEAP assay. And so essentially the addition of tumor necrosis factor alpha, or TNF-alpha, this leads to the activation of NF-kappa B. And the translocation and binding of the AP1 to the promoter leads to the expression of SEAP, which is then spectrophotometrically viewed upon the addition of this colorimetric substrate quanti blue. And so notably from this assay, we could see that the trityl ether analogs reduced SEAP activity in a dose-dependent manner. And also compound 13, the chlorobutyl, actually displayed the greatest dose-dependent decrease across all 4 concentrations. And also one thing to note is that there was no decrease in cell viability in these HEC293s at any of the concentrations. And then this assay, with the CD40 SEAP assay, it functions very similarly to the assay I previously mentioned. Upon CD40 activation with the CD40L ligand, the NF-kappa B pathway is triggered in the cell, allowing for or driving the expression of SEAP. And so in this assay, we saw quite consistent results where compound 13 exhibited dose-dependent SEAP activity. Okay, then in order to evaluate our compound's effect on Wnt1 signaling, we used an in vitro reporter cell luciferase assay. We essentially used stably transfected Wnt1 responsive 3T3 cells that express the TCF-LEF activated firefly luciferase gene. And so we activated this pathway using Wnt3A and then dosed our compounds at 10 and 5 micromolar. And so after treatment for about 24 hours, after the addition of a lysis luciferin substrate solution, we were able to view these results on a luminescence plate reader. And so notably, again, consistent with previous experiments, the trityl ether analogs exhibited potent inhibition of luciferase expression above 5 micromolar. Also, it was previously recognized from that study that C19 analogs show GSK3 beta dependent modulation of the Wnt1 signaling pathway. And so in order to assess whether the activity of our analogs were altered by the direct inhibition of GSK3 beta, we co-dosed these reporter cells with 10 micromolar CHR, which is a selective GSK3 beta kinase inhibitor to activate luciferase expression. And so similarly, the trityl compounds exhibited the most potent decrease in luciferase activity. And so then while trying to address the role of GSK3 beta in any observed viability trends, we co-delivered Andrographolide and the synthesized analogs with this nanomolar potent GSK3 beta inhibitor 𝜷. And we see that at the higher concentrations, there is decreased viability in these colon cancer cells. But also 𝜷 itself does not affect the viability. And also the activity of our analogs from this data we can see bears in HG29 specifically bears greater dependence on GSK3 beta and beta-catenin signaling. And so we then investigated whether the administration of our compounds affects the phosphorylation or localization level of beta-catenin in those specific cell types. And so in order to do this, we performed an immunofluorescence imaging experiment where we stained beta-catenin with a primary antibody followed by an elexiflor secondary antibody, and then counterstained the nucleus with DAPI. And so by overlaying the two images, we would be able to observe the difference between the stained nucleus and the beta-catenin. But however, quantification of these images using ImageJ did not result in any significant changes, allowing us to, I guess, conclude that our compounds do not affect or do not cause a significant change in the beta-catenin distribution, and the difference in the absolute beta-catenin content is small. And so overall, we can suggest that even though beta-catenin is involved in the function of these analogs and their anti-cancer capabilities, the full mechanism involves many more factors just beyond like this one target. But we can confidently say that based on these experiments and this in vitro data, that subtle changes to the C19 alcohol or substitutions at that position cause many changes in the compound's potency and changes to their biological mechanism of action. Okay, I guess this is a quick interruption to my talk about Andrographolide to talk about this other study that was done regarding the Wnt1 pathway and just exploring that place. Again, like our interest in beta-catenin has been approached by both the natural product standpoint with this Andrographolide study and from a small molecule standpoint, which I'll talk about now. So essentially, MSAB is a selective inhibitor of the Wnt1 beta-catenin signaling pathway, and it is known to putatively function through direct engagement of beta-catenin, which leads to the degradation and lowers the nuclear translocation, like I mentioned earlier. But other studies have shown that shifting the ester position would actually change or would lead to comparable beta-catenin inhibitory activity. But there are still questions regarding the direct or indirect beta-catenin engagement or modulation, and their biological activities have not been studied in depth or at length before. And so, in order to understand how changes of this of the identity and the structure and of the methyl ester and how they affect the in vitro potency of this compound in mammalian cell lines, we prepared and evaluated the three analogs of MSAB with 3 and 4 substituted methyl and ethyl esters. We evaluated the anti-proliferative activity of MSAB and its analogs through MTT assays in HCT116, HG29, CT26, MDA-MB231, and HEC293 cells. An example of the cell viability graph is shown on the right. Also, in the table, I have their IC50 values, but I guess I can highlight a few things where, as expected, MSAB exhibited dose and time-dependent potency across most of the cell lines, but compounds 2 and 3, in fact, were observed to have greater dependence on or greater antiproliferative activity only at the 72-hour time point. And I guess this difference in potency in this cell line indicates that the in vitro activity of MSAB is very sensitive to the addition of a single atom or a change in a single atom of the structure, specifically of the ester fragment or repositioning of that fragment from the 3 to 4 position. And so we further wanted to evaluate its potential evaluate the potential pro-apoptotic activity induced by those compounds in a flow cytometry experiment. And essentially a common trend across the cell lines was elevated necrotic cells, which is seen through the PI stain. And the difference in necrotic cells from the control demonstrates the antiproliferative properties of MSAB. And essentially, these elevated necrotic cells in compounds treated with MSAB suggest that changes to the ester fragment is not really well tolerated. Next, to determine the effect of our compounds on Wnt1 signaling, we use this very similar, or I guess the same, luciferase reporter cell assay. And so, consistent with their putative mechanism of action, as Wnt1 pathway modulators, compound 1 and 2, the methyl 3, and methyl 4 exhibited potent inhibition, allowing for more luciferase expression above 25 micromolar, and compound 4 exhibited very minimal activity in this assay at all the concentrations, as it was quite minimally potent, which is similar to or consistent with the cell viability experiments. And so these results essentially affirm or confirm that Wnt1 related mechanism of action of MSAB and related aryl sulfonamides that have been previously studied. Okay, the next part of our study involves synthesizing this moiety on Andrographolide, or this product that has been isolated from a very small portion of the plant Andrographis paniculata. And this material contains the Oxetane moiety highlighted on the right. And its biological activity has not been studied in depth before, noting that it is quite challenging to construct synthetically. And you might be wondering why this small fraction of what is isolated from this plant piques our interest. But essentially, oxetanes are bioisosteres of carbonyls, where these C-double bond O functionalities are metabolic liabilities, especially since they react with biological amines. But oxetanes with conformational rigidity are not as reactive. And so on the left, you can see some examples of natural products that contain the oxetane moiety. And in fact, it is known that much of Taxol's or this compound's biological activity is due to this oxetane moiety. And so we propose two main strategies to construct this oxetane moiety on Andrographolide, one of which is through a C19 nucleophile and a C3 leaving group, and another which is very similar to the other to the first, using a C3 nucleophile and a C19 leaving group. And so these are the two methods that we have hypotheses or are currently focusing on to close the oxetane ring. And currently, DFT, or density functional theory, transition state analysis is being done to further confirm and better understand the conformation and attack that is favored when forming this highly strained ring, this highly strained oxetane ring under varying conditions, some of which that I'll go over later. And so prior to when I myself and some of our current group members have picked up this project, previous group members have attempted to synthesize this oxetane on Andrographolide. And so they attempted using PBr3 after protection at the C19 position using a TBS group. But this reaction led to quick decomposition. And other similar leaving groups were tested on Andrographolide, such as tosyl, nosyl, and bromine, and other bulky groups. And so that's kind of where we started our campaign in synthesizing the oxetane moiety with the C19 functionalization, especially considering that it is the more reactive alcohol. And so they had also tried a series of conditions, a route to synthesize this oxetane moiety. They tried the Appel conditions using CBr4, which was used to give deoxy-functionalization at C19, but the byproduct, which was the dibromocyclopropane, was observed in trace amounts. And a similar set of conditions was also tried with carbon tetrachloride, but this also led to trace amounts of that byproduct, but this set of reactions weren't highly reproducible. And so we realized that the key intermediate necessary in order to perform A-ring functionalization would have to have the C14 alcohol protected, considering the differences in pKa. Essentially, the lower pKa of the C14 alcohol makes it more reactive, and also noting that the C19 primary alcohol would easily get silated. Our goal was to synthesize the C14 protected andrographolide, which could only be possible by first protecting the C3-C19 diol alcohols, and that would only allow for the C14 selective protection. And so we were originally using reported conditions using PBTS and dimethoxypropane, but later optimization brought us to this time-efficient set of reactions or reaction conditions using CSA, where this reaction was complete in just about 10 minutes at gram scale. And so following this ketal protection, we needed to decide on a silicon protecting group for the C4 to protect the C14 alcohol. And so we tried dimethylthexyl, which unfortunately was quite sluggish, but then got some success with the TBS and TES protecting groups. But later on, we were also able to find that we were able to get selective ketal deprotection with triethylamine hydrochloride, but these conditions were quite sluggish and not very convenient to perform at large scales, which is what we wished. And so we ended up choosing this set of conditions with the TIPS protecting group at the C14 position. That is quite strong of a protecting group that we would later be able to deprotect when necessary using TBAF. And so I guess to organize this approach, or what I have just said, we were able to achieve this C14, this intermediate, by essentially protecting the like with the same conditions I mentioned before, with the acetonide protection followed by a C14 TIPS protection, and then a formic acid or any acid deprotection of that ketal, giving us that valuable C14 TIPS intermediate. And so using this intermediate, we were able to screen a set of leaving group conditions, such as the triflate, the mesylate, the thionyl chloride, and trifluoroacetic anhydride. And we found that the triflate leaving group, this reaction was quite temperature and reaction condition sensitive, and therefore was not very sufficient to perform at larger scales. The thionyl chloride is a reported reaction that, as expected, would work, but was quite slower than the recently founded TFAA as a reagent to construct this Oxetane moiety. And this produced this reaction had quite clean conversion, and so we might be able to perform this reaction at large scale if necessary in the future. And so we were simultaneously performing C3 functionalization. So we used our C14 TIPS key intermediate and protected the C19 alcohol, leaving the C3 alcohol open for functionalization. And so we tried direct functionalization of the C3 alcohol with conditions listed or highlighted in the table, but we quickly realized that in order to construct the Oxetane moiety in the best way possible in the correct stereochemistry, we would need to invert the C3 alcohol stereochemistry. And so this led us to, or I guess we're now planning to attempt this route to reverse the stereochemistry of the alcohol, first by oxidizing it to a ketone and then performing an anti-selective reduction such as the Evans-Saxena. That gives us the opposite face of the alcohol, and we would later test conditions, leaving group conditions, in order to close that Oxetane ring. So to conclude, on the top, we've highlighted some compounds that we have made so far and characterized, including key intermediates that would allow for future diversification and future, I guess, library of analogs. On the bottom shows a few analogs that we're currently trying to make. And I guess in the future, we'll expand this library to include many other functional groups on the A ring, including some cyclic ones as well. And we will also attempt or further explore the DFT transition state analysis in order to find the optimal conditions to synthesize some of the analogs in addition to the oxetane. And hopefully we'll be able to test some of our analogs for their biological activity as well. Okay, and these are both of the papers that we have out. The first one, Andrographolide, is in Bioorganic Medchem Letters, and the second is currently in bioRxiv. Yeah. Okay, and then I'd like to thank, obviously, my advisor, Dr. New, and my lab members who have helped me incredibly throughout this process, in addition to some of our collaborators. Okay, thank you. Do you guys have any questions?